First off, welcome back. Hope everyone had a lovely, restful vacation. Restful meaning from schoolwork. Sort of. Not really. I know swimmers were busy swimming, right? Softball and baseball players were soaking up the sun. I'm not jealous. Maybe a little, maybe a lot. But at any rate, we're back. And usually after spring break, it is a very quick progression towards the end of the semester. All right, we basically have 5 weeks left. All right, you have an exam on Friday, correct? The 28th, right? Okay. So, exam on Friday. We'll cover everything from the last exam through Wednesday. Okay? So, we'll go over the material that covers again on Wednesday. And I will post a study guide probably tomorrow. Okay? Tomorrow. Questions on that? Exam format the same, multiple choice, etc. Okay? All right. Second item of business is that you will have your midterm grades posted. All right, I'll post them tomorrow. They're basically based upon your quiz grades and your first exam. Okay, because your second exam, obviously, you won't take until Friday. Okay? So, midterm grades will be posted by probably on Tuesday. Okay? All right. So, from that point on, we have to go on to the next. When we left off before spring break, we finished with 16.1, which was talking about differential gene expression in terms of development. All right? And when there's changes in developmental gene expression, it can lead to very big changes in the final outcome, shape, structure of function of the overall organism. And what organism do we talk about in relation to that, or do we use an example for that? What organism do we use? We talked about the frog, and we also used the fruit fly, right? Drosophila. Okay, the fruit fly, and we went through segmentation. We introduced homeotic genes, okay, as being important for identifying axis formation, axis determination, top, bottom, left, right, front, back. Okay, and then we also said there's changes in terms of what the boundary is, beginning of the end of the head, beginning of the thorax, end of the thorax, beginning of the abdomen in terms of the fly parts. And then we also went over one example of a gene or a gene and a protein product of that gene that's important in directing axis determination. And what was that product? What was that gene/protein product? Bicoid. All right, and we said bicoid was a morphogen and depends upon a concentration gradient from the most concentrated being the back end, the least concentrated, or vice versa. All right, and there's a couple other similar morphogens that are involved in that. Okay, so the idea about chapter 16 is gene expression and how it relates to development. Okay, and the next part of this is we're going to talk about cloning and stem cells. Okay, because stem cells are dependent upon gene expression to identify and differentiate into all the different cells of the body. All right, but first, cloning was used to show that differentiated cells could be reprogrammed. All right, and you'll see this in a little bit. All right. Now, when I say cloning, what do you think about? What's your word association? Cloning. What are you thinking about? Dolly the sheep. Right? What else?
Like just a double
So you're like identical twin. Okay, so that's in and of itself is reproductive cloning. All right, so you're going to take and Dolly the sheep, and we'll talk about Dolly in a little bit, a special technique that's used to use a nucleus from a differentiated cell and then put that into a enucleated egg and then having that become a new organism. That is reproductive cloning. Okay? So in this case, we're going to talk about, there's a couple different, there's molecular cloning, right, which talks about, involves DNA sequences and DNA parts. And then there's organismal cloning, and then there's reproductive cloning. All right? Organismal cloning, basically, one or more organisms develop from a single cell without meiosis or fertilization. And this happens in nature. Okay? It is a process called parthenogenesis. Parthenogenesis. All right? Which is this. Parthenogenesis. Whew. It's a long word. Parthenogenesis. Okay? Parthenogenesis is the process by which frog eggs, okay, can actually begin and develop without being fertilized. Okay? It is a spontaneous development of a meiotic or a haploid cell. Okay? Typically an egg. Okay? So in this case, one or more organisms develop from a single cell without meiosis or fertilization. Right? So parthenogenesis is an example of organismal cloning. Right? That happens naturally. All right? When that occurs, your cloned individuals are obviously genetically identical to the parent. All right? At least in the haploid form. And then, but organismal cloning more recently is obviously used for medical, really interest leads in medical potential to generate stem cells for medical therapies and regenerative medicine. Okay? So really we're going to concentrate on this in terms of humans and mammalian organism development. Oh, thank you. Colds suck. So Emily mentioned Dolly the sheep, all right, which you guys, being born in, 1994, right, 94, 95. You were 2 years old when this happened. I was in college, okay? So I was sitting where you are, okay, learning about molecular biology, and this Dolly happened. So you've sort of grown up with the idea of stem cells. It was sort of, for me, a new area of research that I've sort of grown up with. But the idea of cloning is even older than that, okay? So cloning in plants is a little easier. You think you sort of do it a lot, all right? Any of you ever have taking like a clipping or a piece of a potato? You ever grown a potato plant from a potato itself in elementary school? No? God, they don't do science at all in elementary school. So it's easy. You go to the grocery store, right? You buy an organic potato. You go home. You cut it in half. Okay? You put a couple toothpicks in the side so it doesn't sink submerge it completely in water. And a potato is a root. It will grow. Okay? There what we call eyes off the root segments will grow off the potato, and eventually you will get potato greens, and you'll have a potato plant. You can grow a potato from a portion of the potato. That in and of itself is cloning. Okay? You've basically cut open and expose the stem cells within the root that have now become a new plant. You do that to say you cut the potato in 4 sections. Now you have 4 potato plants instead of 1 that are identically correct, identically the same. I mean, this has been happening for years, right? For hundreds of centuries, people have done this. They don't know what they're doing. They just take all right, it's a root, let's split it up. Well, in 1950s, Stewart and it went sort of the next step. Well, what part of the carrot, another root vegetable, actually will isolate cells from that carrot, and let's see what cells can become a new plant. All right, so we've taken the next step. So by this work, he looked at single differentiated cells from the root, incubated in a certain media, all right, were able to grow into complete adult plants. All right, so in this case, it showed that differentiation is not complete, necessarily irreversible. All right, this was sort of lays the groundwork for the potential of reprogramming cells, all right, even differentiated cells. And then also developed the idea that some cells can become all the cells of the body, and other cells sort of are fated to become a certain type of cell. All right? So cells that can give rise to all the specialized cells types in the organism are called totipotent. All right? Potent refers to potential. Toti sort of sounds like which word? Total. Right? So they have total potential. They can become anything at that point. All right? So embryonic stem cells are totipotent. All right? They can think about the cells of the developing embryo after fertilization. They have to become, right, or be able to become neurons, muscle, skin, bone, kidney, whatever you're looking at, right? So they are totipotent. So, in terms of Dolly, cloning of animals, which first happened in like 1997, all right? What occurred was that the nucleus of an unfertilized egg was replaced by another nucleus of a somatic, what we call a somatic cell, a skin cell, muscle cell, whatever, right? So a body cell, a non-germ cell, not a sperm cell, okay? This is what's called nuclear transplantation, right? What we call somatic cell nuclear transplantation, all right? Somatic cell nuclear transplantation. Now, this was first done in frogs, all right? So experience with your frog embryo showed that a transplanted nucleus can often support normal development of an egg, right? So you transfer the thing. This was first done in frogs. Why? They're less complex. What else happens spontaneously to frog eggs? Parthenogenesis, right? So the idea of some frogs' eggs can spontaneously sort of develop on their own. There has to be something possibly involved in the frog biology that could allow this to happen. So let's see what occurs. I mean, because basically, you're looking at sperm egg fertilization. What is the sperm carrying? The sperm is a glorified nucleus, right? It's just genetic material with a motility action, a way of moving, okay? So stripping away sort of the motive or the motive force of the sperm, you're looking at somatic cell nuclear transfer. You're actually just transferring a nucleus, all right? Now, The older the donor nucleus, the lower the percentage of normally developing tadpoles. All right, so age has something to do with this. All right, why? What in terms of biology you think might have to do with this, Abby?
Like depending on how differentiated the cell might be.
How differentiated the cells might be? Exactly. All right, so how long they've gone in terms of terminal differentiation, and then how many rounds of mitosis have that happen? All right, those cells get older. What happens at the ends of chromosomes as cells get older? All right, the telomeres shorten. All right, so that has something to do with it also. All right, less fidelity of replication. All right, more errors that are prone. It can also have something to do with it. All right, and then Gordon concluding from this work that nuclear potential is restricted as development and differentiation proceeds. All right, so this is as a embryo develops 2 cells, 4 cells, 8, 16, 32, and so on. At some point, those cells of that embryo lose their totipotency. They go down a certain pathway to differentiation, and they can't go back. Okay? That's sort of the restriction. So here's your experiments with your frog embryo. All right, so a less differentiated cell, donor nucleus is transplanted. Most tadpoles develop into normal tadpoles. All right, so let's say you take a frog embryo and you take one of those cells, put it in from an embryo and put it into an egg. It develops on its own. Okay, but now you take a fully differentiated intestinal cell. Okay, take that nucleus out, put it into an egg. They only develop so far. Which one of those cells is less differentiated? Which one of those starting cells, the intestinal cell or the embryo cell? Or the embryo cell. Which one is totipotent? The embryo cell, right? And the only thing that the intestinal cell can be is an intestinal cell. But there's some reprogramming that's occurring within the donor nucleus, within the egg. When you transfer this nucleus over, there's some reprogramming that's going on that allows genes to be produced for some normal development to occur to a certain point. Okay? Questions on that? Now, years have gone by. All right, looking at cloning, the possibility of stem cells. In '97, Ian Wilmot, all right, and his colleagues announced the birth of Dolly, a lamb cloned from an adult sheep by nuclear transplantation from a differentiated cell. Somatic cell nuclear transfer was born. Okay? Normal, developed lamb, completely normal in terms of biology, shape, structure. When she came, normal birth, was born, right? Normal development. Until a certain point. As they fought, what's good about sort of having a normal development organism, they were able to follow her development and lifespan over the next few years. She died prematurely in 2003, so a 6-year-old lamb, or 6-year-old sheep by that point. That's pretty short lifespan for a sheep that's well taken care of, right? And that's not, say, for meat gain or for meat use or anything, right? Well taken care of in captivity. Usually sheep can live 10 to 14 years. Okay, so 6 years is sort of premature. She had arthritis, all right, the premature development of arthritis. What does this tell you? What conclusions could you make from that?
Her cells were older, kind of.
Right, so maybe she wasn't, her health wasn't as, she wasn't as fidel, or she doesn't have the fidelity or the vitality as a normal sheep born from, normal fertilization would have been. All right, possibly reflecting some incomplete reprogramming. All right, incomplete reprogramming from above the original transplanted nucleus. Still, this showed the possibility that complete, at least reprogram to form new individuals can be done from a somatic cell. All right, and this is what happened. They took mammary cells from the mammary gland of a sheep. Okay, an egg from another sheep. So really, they're two separate genes, or two separate. Well, obviously female, right, but two separate genetic backgrounds. They cultured the mammary cells, right, so to produce clones, and then take one of those cells. They were arrested in the cell cycle, causing them to differentiate, and then take that nucleus, put it into the egg. Okay, and then you end up. There was a certain type of procedure that was done to induce the development of the egg itself into an embryo, early embryo. They implant those embryos into a surrogate mother. One implants, attaches, develops, and then Dolly was born later, with genetically identical to the mammary cell donor. All right, so this nucleus was removed. All right, it's important that they didn't. Why was it important that they used two different genetic backgrounds? Why use so your two female sheep were involved at the beginning of this. All right, one is going to be the donor, the egg donor. One is going to be the source of cells. Why use two different sheep? Why not just be the same?
So you can make sure it was cloned of the mammary cell.
So you can genetically compare at the end, that if this works out, that the cloning was a success. Okay? And then this is a third sheep. Okay? Genetically different, so that you can distinguish and go back and prove that the Dolly came from the cultured mammary cells. All right. Since 97, cloning has been demonstrated in other mammals, mice all the time, cats, cows, horses, mules, pigs, dogs, lots of different. Since then, there's been more. Okay. All right. CC for carbon copy was the first cat cloned. Right. However, CC differed somewhat from her female parent. All right. You'll see this in a second. You'll see that cloned animals do not always look exactly the same. All right. Why? Why might this occur? Especially in cats. What happens in females that can alter their appearance? That doesn't necessarily happen. Let's say female identical twins that can alter their clones that can alter their appearance. I'll show you. Here's Cece here. There's a picture of parent and Cece right there. Do they look different? All right. Genetically, they are identical. Why do they look different?
Gene expression. Which genes are expressed?
Gene expression?
Because isn't it for cats like one gene has to be on for the coat color?
All right, so one gene, so coat color especially in cats is important. All right, one's on, one's sometimes off, depending on which cell you're looking at. Okay? What is occurring in those cells that will shut off gene expression? especially because they're females. The X chromosome. All right? Because females have two X chromosomes, and genes on those only one of those X chromosomes is active at a certain time. In terms of cats, coat color is an X-linked gene. Okay? So in some cells, one X chromosome can be shut off, and the other one could be turned on. That is a complete, researchers believe it's a non-random process, but it is not the same between individuals. Okay? So although researchers believe it's a non-random process, there's no way of sort of predicting it between two genetically identical individuals. So here you have clone copy of genetically identical cats that look completely different based upon their X chromosome inactivation, gene expression. All right, this is not an efficient process. It is not meant to be an efficient process because naturally you're supposed to have some genetic mixing of genetic material in order to produce a viable individual. All right, so as evidence for this, the inefficiency of cloning is due to faulty genes. Okay? So in most of these nuclear transplantation studies, a very small percentage of cloned embryos have developed normally to birth. All right? So even when we go back to Dolly, there were many embryos implanted into the surrogate mother, and one of them actually grew. All right? You think about reproductive fertility treatments for humans now. Okay? They do in vitro fertilization, and they implant several embryos. Why? I mean, even in vitro, this is a sort of a natural thing that's occurring, but they transplant several embryos into the mother and hoping that one, maybe two, implant and become successful. All right, because it's an inefficient process. All right, many cloned animals exhibit defects, and this may be related to epigenetic changes. In order for the cell to develop normally, there has to be some reprogramming. DNA, it has to be demethylated. Epigenetics sort of information has to be taken off and removed in order to start over again. That's sort of the reprogramming process. All right, so the epigenetic changes must be reversed in the nucleus from a donor animal in order for genes to be expressed or repressed appropriately for early stages of development. That, in a nutshell, or at the sort of the basis, is the reprogramming process. What is happening in cells that's going to remove the X chromosome inactivation, remove DNA methylation, allow those genes that have been shut off during differentiation to be turned back on. All right, so that they can become a normal. All right, so then this brings us to the idea of stem cells. All right, a stem cell is basically defined as a relatively unspecialized cell, undifferentiated, that can reproduce itself, right? So a stem cell has the idea of self-renewal. It can undergo mitosis and form another stem cell, and also differentiate into specialized cells of one or more types, okay? And there are several different types of stem cells. In your body, right now, there's red blood cells are being produced. White blood cells are being produced. Okay? They're being produced from a stem cell. Red blood cells and white blood cells originate from a similar or the same cell, a hematocytoblast, otherwise known as a hematopoietic stem cell, right? Which can give rise to all the cells of the blood: white blood cells, red blood cells, and thrombocytes. Platelets. All right? That is a pluripotent stem cell. We'll go over pluripotency means what? So if a hemangiocytoblast can become a white blood cell, including neutrophils, basophils, macrophages, monocytes, platelets, and a red blood cell, it can become one of 6 different cells, right? Okay. So, but can it become a skin cell? No. It can become multiple types of cells, but not every cell of the body. So it doesn't have total potential, but it has multiple potentials, right? So plural, pluri, it can become many types of cells, all right? Cells isolated from the embryos at the blastocyst stage are called embryonic stem cells, all right? These are able to differentiate into all cell types. They are totipotent, okay? The adult body has stem cells to replace non-reproducing specialized cells. So every day you are losing cells to the environment. Your skin, dead cells are in the skin. You have epidermal stem cells that become the epidermis, that regenerate the epidermis. Skin, you have muscle, myoblasts that produce muscle cells as necessary, and so on. All right? So they obviously exist as well. All right? This is the idea of a stem cell. All right? Important is self-renewal. Okay? The stem cell has to be able to produce a copy of itself so that the stem cell population is still there and can continue to produce these other cells. All right? So this is the first step of cell fate. Cell fate. All right, so this cell is now fated to become a fat cell, bone cell, or a white blood cell, depending on where it's going. All right, as an example.
So what is sulfate?
It's like a determination.
Okay.
All right, so in this case, this cell is determined to become, this precursor cell is determined to become one of the other cells. It is no longer, it's lost its stemness. All right? This is comparing embryonic stem cells versus adult stem cells. What do you, looking at this graphic, what do you see?
Adult stem cells depend on their location for something. Okay.
Sort of. So adult stem cells depend upon their location for cell fate. Ben?
Adult stem cells can only regenerate or grow to a certain type of cell.
So they've been fated. Okay? So they've lost their totipotency. They've been faded a little bit to only become one of certain types, right? So the embryonic stem cells can become any of these, right? Liver, nerve, muscle, and also blood. But the adult stem cells, right, you look at the hematopoietic stem cells that they've isolated here can only become blood cells. Right? They can't differentiate. Now, there has been some research into sort of reprogramming cells to become going from here to, say, there, right, from adult stem cells. And we'll get into that in a little bit, right? But in the normal body, aside from outside intervention, embryonic stem cells are totipotent, while adult stem cells are pluripotent. All right? This is sort of not true. Why? Embryonic stem cells are totipotent. All right, depending upon which stage of the embryo you take it from. But if you get cells prior to their original cell fate, their first cell fate determination, they are totipotent. All right, so you could just sort of cross this out, and you could say, stem cells. And I'd say adult are pluripotent, capable of differentiating into many cell types. All right? Now, another major thrust of research has been, probably in the last 7 or 8 years, has been the idea of taking a somatic cell, which has all the DNA necessary for same DNA genome that a stem cell has, right? So on one side you have this cell, nucleus, right? In terms of DNA, these two cells are not different, right? If this one is a stem cell, and this one is a somatic cell, okay? Fully differentiate. In terms of DNA, the genome, they come from the same person. They are not different. They have the same genes. What is different between them? Right. Level of differentiation, which is a result of what? Gene expression. So the only difference between these cells, at the heart of the difference of their behavior, their biology, is gene expression. Okay? So gene expression. And prior to this, it's been shown that you could take this cell nucleus and make it behave like a stem cell if you put it into an egg, right? That's the idea of somatic cell nuclear transplant. You can produce a whole new embryo and a whole new organism from that process. In order to do that, there's some reprogramming that occurs, right? Of the genes inside that somatic cell, that non-stem cell, that makes it behave or allows it to behave like a stem cell. Correct? So now the question was, is there a way, or are there genes that we can turn on in the somatic cell that can make it behave like a stem cell? Can we take a fully differentiated cell from the skin, for example, reprogram it somehow, and now it behaves like a stem cell? And what they did was they used viruses, okay, retroviruses, to reprogram the cell and turn genes back on that were important for stem cell behavior. In a way, they're what's called inducing stem cell. All right, so use these retroviruses, and it basically got down to, one of the first papers was taking a whole set of genes that are important for stem cells, were important for the identity of stem cells. So stem cells are lineage negative. This gene's off, that gene's on type thing. Okay? So they took that sort of genetic profile and put a bunch of those genes back into a somatic cell, and then it started to become like a stem cell. This is the idea of what's called an induced pluripotent stem cell, or an IPS. Okay? So you could induce a cell to act like a stem cell. All right? Over time, they've gotten it down to about 4 genes that are important to turn a cell back into a stem cell. All right? And these are actually genes of transcription factors that are usually shut off during development. All right? OCT4, NANOG, they're important, and they regulate the expression of a whole host of other genes that are important for stem cell regulation or stem cell viability. All right? what advantage does an IPS, an induced pluripotent stem cell, have? What do you think it could be used for? What's the big idea? Let's put it that way. Regrowth. This is so what's the controversy with using stem cells, embryonic stem cells, for regenerative medicine? Where do we get embryonic stem cells from? Embryos. Kind of an ethical problem there, right? Big problem. Okay? For years there was no embryonic stem cell research because of religious, moral, moral discrepancies, moral problems with embryonic stem cells. Okay, this procedure sort of takes away that moral dilemma. Now, Nick here ends up with kidney disease. Not wishing it on you, Nick. Say you do. You have to go on dialysis. What's the only way to sort of replace kidneys? Is a what? Transplant. Kidney transplants are moderately successful if you find a match, right? But from that, you also have to take anti-rejection drugs for the rest of your life, etc., which run on the order of between $5,000 and $12,000 a month for the rest of your. Yeah, that's not necessarily covered by insurance. Okay, so it's a big deal, and then you also run the risk of rejection. Okay, but what if the idea being, I take one of Nick's skin cells, do the procedure of inducing it to become a pluripotent stem cell, and then differentiate that cell, those cells, into kidney, and then grow a kidney organ on a scaffold so that it becomes all the different parts of the kidney and it works as a kidney organ. What is the, well, I mean, that's a big idea. That's a long way away. But that's the goal. Now, in terms of rejection, do we have any problems? No, it came from him. Okay? He still has all the same genomes. A genome in all those cells are the same. But now you reduce that amount of rejection. All right? Now you have organs that can be produced. Okay? That's sort of a grand idea, a grand sort of scheme of down the road. More likely is that they use them for more cellular therapies. All right? Patient's own cell may be reprogrammed, right, to potentially replace the non-functional disease cells. All right, and this is probably one of the problems of this. One of the probably more near future ideas is in terms of diabetics. Okay, diabetics have reduced production of insulin. That's produced from where? The pancreas. All right, specifically the beta cells of the pancreas. For whatever reasons, type 1 or type 2 diabetes, type 1 is genetic, type 2 is sort of comes on as time goes on. All right, but for type 1 diabetics, they don't produce insulin. Okay, so you take an IPS cell, reproduce it. You take a somatic cell, induce it to become a stem cell, and then differentiate it down the line of a beta cell, inject those cells into the pancreas, and they become insulin-producing beta cells. All right? That's not even to talk about what you can do to the cells outside of the person to change the way that they behave. Have them produce different genes, GFP, whatever it might be. Okay? Higher amounts of insulin, or a better type of insulin, whatever it might be. All right? But that's sort of the goal of regenerative medicine. And now with this, the idea of, or the ability to produce induced pluripotent stem cells, you remove that ethical problem of embryonic stem cells. All right? To this date, they have not, IPS cells are still not as viable as embryonic stem cells, though. All right, there's some more research that has to be done on that front. Question on what a stem cell is and its potential uses. On Wednesday, all right, we will talk about another aspect of gene expression that is changed in the human body, cancer. Okay? So, till then, I will see you on Wednesday.